Hello. Today we're going to talk about my favorite blood group system, the Rh system. It's one of the most complicated systems. It has 56 antigens as of today, and it has a very convoluted history, which we'll talk about a little bit. Like many blood group systems, the discovery of the Rh system started with a patient who developed antibodies. Now, the Rh system has 56 known antigens. In 1939, Levine and Stetson reported a case of hemolytic disease of the fetus. The child was a stillborn and the mother needed a blood transfusion. The husband donated blood and the mother had a hemolytic transfusion reaction. Levine and Stetson performed additional testing with her serum and found it agglutinated 80% of ABO-compatible donors. Around the same time, Landsteiner and Wiener were performing experiments by injecting monkey cells into rabbits. They found the rabbits' serum agglutinated not only monkey cells, but also 85% of human RBCs. They called this anti-rhesus antibody after the monkey. It wasn't until 1962 that it was determined by Levine that Levine and Stetson's antibody was not the same as Landsteiner and Wiener's. The human antibody was finally named anti-D, and the rabbit antibody was named anti-LW after the scientist. Fisher and Race proposed the three-gene theory in 1943. They thought D, C, and E were the main genes. Crossover is rare, but some antigens are produced due to the combination of genes. In 1943, Wiener proposed that there was only one gene associated with the Rh system. And in 1986, Dr. Tippett proposed a two-gene theory. This is still believed to be true. Fisher-Race and Wiener are the two systems of nomenclature before molecular techniques were around. There are two additional systems now, but Fisher and Race and Wiener are still used frequently, and you should be able to understand what they mean. We'll go over them a lot more on the next couple slides. So remember, Fisher and Race believed there were three genes. First gene being D, so you either had the D antigen or no D antigen. There is no little d antigen, remember this. Usually in homework or when it's written, people might put a little d just as a placeholder, but there is no D antigen. There is either D or no D. The second gene is C, so you can either have a large C or a small c, and then same with E, big E or little e. So even though Fisher-Race believed there were three genes, they did also believe that they were inherited as a haplotype. So you got all three genes together from one parent. Likewise, Wiener believed that you got all together from a parent as well, but they thought it was one big gene. Instead of a haplotype, they thought it was just one big gene. So they had 8 different allele names, which have been modified because the original terminology was very confusing. The modified versions are RO, R1, R2, RZ, little r prime, little r double prime, and little ry. This is just a recap of the nomenclature. We went over Fisher-Race. This is old Wiener terminology, what he originally proposed, which has been modified to an easier way to discuss it verbally. So this is a very important slide. I would study this until you are completely comfortable switching back and forth between Wiener and Fisher-Race terminology. So to start, Remember, Wiener believed in one gene. So in his terminology, you get the whole haplotype in one allele. So Fisher-Race believed there were 3, so he kind of spells it out in his terminology. So you'll have it written out for each antigen what it is. So how we kind of break this down and remember it. The D equals R in Wiener terminology. So if you have a big R, you have a D antigen. If you have a little r, there's no D present. For the CE, remember that little c, little e is the reference allele. So the numbers or letters are basically telling you what's different from the reference allele. So if you have RO or little r, just think there's 0 changes from the reference allele. So a big R, O will be a D and a little c and a little e. 1 means big C. So if you have R1, the big C is different from the reference allele. If you have 2, the E is different from the reference allele. So you have a big E. And then for the RZ and the RY, these are very rare alleles. You don't typically see these. It's very low percentage, but that means both are different from the reference allele. So I would just practice switching back and forth until you really understand. Because in the blood bank, we do use this terminology a lot. We actually organize our units according to if they're R1, R1, R2, R2, RO, RO. So you should know this terminology. And this is just a list of frequencies. Like I said, the RY and the RZ, very rare. But it is different based on race. So it is good for you to memorize which ones are the most common in each of the races. Next, we have Rosenfield terminology. This is completely unrelated to any of the inheritance theories. It is simply each antigen, is it there or is it not? So you would write this RH colon and then 1, 2, 3, 4, 5. If the antigen is there, you're not going to have a minus symbol in front of it or a hyphen. And if the antigen is not present, then you would put a hyphen in front of it, meaning it's not there. This is pretty easy to read, but it's not so great for verbal communication. If you can just think of telling someone, oh, I need a unit that's RH colon -1, 2, -3, -4, 5. No one has time to sit around and say that out loud. Saying R1, R1 is much faster and easier to understand. And the last of the terminologies is ISBT. It is a 6-digit number. So when we're talking about ISBT, the first 3 numbers are always the system number. RH system is system number 4. And then the remaining 3 numbers represent the specific antigen that you're talking about. So, for example, D is antigen 1. So if you spelled this out, it would be 004, 001. Also, not something you're going to say out loud. So ISBT terminology also has shorthand to make things even more complicated. So for the D antigen, you could also call it RH1 or 4.1, and so on. This would be a good system to use in a computer system to keep everything organized. But again, most people aren't going to be speaking this out loud. And here's another little cheat sheet for you to kind of read through until you can really nail down this nomenclature. So you can just read over this slide, make sure everything makes sense. So again, to go over genotype versus phenotype. Phenotype is when you are actually testing with Antisera, typing the patient or donor's cells. You cannot know the genotype with certainty based on the phenotype. but we can use that frequency data to kind of guess what the haplotype present is. So I really love this image because it shows the two genes, the RHD and the RHCE genes. You can see they're very close together. So the green blocks are the D antigen genes, and then the white blocks are the CE antigen genes. So you can see there's a lot of crossover that makes these strange alleles. There's some where the D antigen is transferred over into the CE gene, and then there's some where the CE gene is inserted into the D gene. And then to make things even more complicated, we also have the RHAG gene. So this is a gene that is necessary for Rh expression. However, it doesn't actually make any antigens. It just makes a protein that's needed for the other Rh antigens to be expressed. The RHAG gene is not polymorphic. The protein forms a tetramer composed of 2 RHAG molecules and 2 either RHCE or RHD molecules to form a core complex. RAG is called a co-expressor because it must be present for successful expression of the Rh antigens. The RHD and RHCE encode the RHD and RHCE proteins. They are direct gene products. The Rh antigens are carried on 3 non-glycosylated transmembrane proteins that are encoded by only 2 genes, RHD and RHCE. Alternative mRNA splicing is probably responsible for the production of 2 distinct polypeptides from the single RHCE gene. Products of both RHD and RHCE are proteins. They traverse the red cell membrane 12 times, displaying short loops of amino acids on the exterior. Deep positive individuals can be homozygous or hemizygous for the presence of the RHD gene. The D antigen is the most immunogenic antigen in the Rh system. The concentration of D on the cells varies with the other antigens inherited. So if you recall back to that image that showed the RHD and RHCE genes, if you look back, the D dash is when the D gene takes up most of the CE gene. So it makes sense that D dash phenotype has the most D antigens on the cells. How do we get a D negative phenotype? In Caucasians, the D negative phenotype almost always results from homozygosity for the deletion of the entire RHD gene. However, in D negative Black populations, they usually have an intact RHD gene, but the gene is inactivated. There is a gene called RHD-Psi, which is a pseudogene that produces no D protein and no D antigen. The Rh-null phenotype produces hemolytic anemia. You'll see stomatocytes in these patients, and you'll see compensated anemia. There's a couple different reasons this phenotype occurs. They call the first the regulator, and then the second an amorph. In the regulator, the RAG gene is mutated or deleted, and in the amorph, the RHD is deleted, and the RAG expression is reduced. There's also the RH mod. In this, the RH protein and the RAG proteins are both reduced. So the D deletion phenotype is the one we spoke about before, the D dash dash, where the D gene actually deletes the CE gene, making the CE non-functional. So there will be no CE antigens present, but a stronger D antigen activity. Then we have weak D phenotypes. Weak D phenotypes have a reduced concentration of D, but they still have all epitopes present. there's a few different reasons weak D phenotypes exist, which we'll talk about on the next slide. Weak D was formerly known as DU. Do not use this terminology, it is outdated. So genetic weak Ds cause a quantitative difference, not a qualitative difference. So all the D epitopes are present, so they should not make anti-D if exposed to D positive blood. It's usually caused by a single amino acid change. Weak D expression can also be caused by a positional effect. This is also called the Cipollini effect. It is when the big C antigen is inherited in trans to D. The steric arrangement of the C interferes with the expression of the D. They are also not at risk for developing an anti-D, so they can be transfused with D positive blood. Then we have partial D phenotypes. These phenotypes are missing some of the epitopes of the D antigen, so they can make anti-D if they're exposed to normal D positive blood. Only pieces of the D are missing. This also used to be called D mosaic, but that's also not a term that's used anymore. How do we treat partial D patients? For pregnant females, they should receive the RHIG injection so that they do not create anti-D. And for adults or children, they should receive either Rh negative RBCs or genotype matched RBCs. If we give genotype matched, we can be certain that they have the donor has the same epitopes present that the patient does. So then they won't be at risk for making antibodies to the other epitopes that they don't have. Weak D testing. For donors in the blood center setting, all donors must have weak D testing performed to verify that there are no D epitopes available. We don't want to give a weak D or a partial D to a D negative patient. For patients, weak D testing is not required except on babies of D negative infants, and that is to know if the mother should receive RIG if they are at risk of exposure. Back to genetics. RHCE has greater than 80 known alleles. Variant forms of the C and E antigens are generated by base substitutions in the RHC gene. A single G to C transition in exon 5 of RHCE results in a polypeptide that reacts with anti-little e rather than anti-big E. Alleles of RHCE that express the C antigen differ from those expressing the little c antigen at 6 nucleotide positions, 1 in exon 1 and 5 in exon 2. Only 4 of these result in amino acid substitutions. This slide shows some of the other Rh system antigens. There's F, which is only present when little c, little e is inherited cis. There's CW, B, and G. The Rh system has many compound antigens. These antigens are defined by their antibody, and their antibody cannot be separated. F is one of these. It is present whenever C and E are inherited in cis. There's also big C, little e antigen, which is also known as Rh7. So how do we know if F is present? If C and E are inherited on the same chromosome. So you can see here, if they have little c, little e on the same chromosome, they will be F positive. However, if the little c and little e are inherited on different chromosomes, they will be F negative. There's also a big C, big E antigen called Rh22. This is when big C and big E are inherited in cis. And then there's Rh27, which is little c, big E. The G antigen is an antigen that stimulates production of an inseparable anti-CD antibody. The G antigen is present on any cell that has C or D or both C and D. Anti-G is a clinically significant antibody, so to avoid a transfusion reaction, we would transfuse with B negative, C negative units. However, in pregnant women, we would need to figure out if it is an anti-G or an anti-D and anti-C to determine if they need RIG or not. Rh antibodies are not naturally occurring. They need a sensitizing event from either transfusion or pregnancy to stimulate production. Most antibodies are IgG. Some may have an IgG component, so they usually react at 37 degrees. They are increased by enzyme treatments, and they often activate complement. So remember, the D density varies based on the rest of the Rh phenotype, so anti-D does react stronger with R2 cells. Rh antibodies are also implicated in warm autoimmune hemolytic anemia. And though they are common not naturally occurring, there have been very few cases of naturally occurring Rh antibodies. Again, Rh antibodies are enhanced by enzyme-treated RBCs. So if you have an antibody that's reacting a little bit weaker that you think might be an Rh antibody, you can treat the RBCs with Fisin or papain, and that should bring out the reaction a little bit stronger. Anti-big E is the most common Rh antibody. Anti-little c is the most important after anti-D. They can cause hemolytic disease of the fetus and newborn. Most autoantibodies in warm autoimmune hemolytic anemia are directed against antigens in the Rh system. Auto anti-little e is the most common. So remember everything we learned about antibodies. That's going to translate to how we deal with transfusions. Most commonly, you will give D negative blood to D negative patients. However, in emergency situations, if D positive blood is the only thing available, we would triage based on age and gender. So we might give males or women above childbearing age the D positive blood if that's all that's available. So that's all for the Rh system. Moving on to Landsteiner-Wiener system. It is ISBT16, it is on chromosome 19, and there are 3 alleles, LWA, LWB, and LW. LW is an amorph, so if you inherit the LW gene, there will be no LW expression, and you will phenotype as LWA negative, B negative. The Rh antigens must be present before the LW antigens can be expressed. So an Rh null person will always be LW negative. Auto anti-LW is also common in patients with warm autoantibodies. Antibodies to the LW system are rare, but they are clinically significant. Anti-LW will often look like an anti-D when you're doing a panel. So how do we differentiate? The LW antibodies will react stronger with RhD cells, and they may not react with RhD negative cells. The antibodies will not react with DPT treated cells, and the antibodies will react very strong with all cord blood cells, D positive and D negative. They also will not react with Rh null cells. And we're finally done. I know the Rh system is so complex and a little bit intimidating, but jump in there and get very familiar with the terminology and have fun.